Thank you very much, Sam, for agreeing to give a Build-a-Cell seminar, and you can get started.
Okay. Well, hi, everyone. Thank you for coming. My name is Sam Butcher. I'm at the University of Wisconsin-Madison. And title of my talk is Build-an-RNA with 2 nucleotides. And I thought, I know this sounds a little strange, but I thought I could maybe gamify this for people. And so here's the game. You get 4 nucleotides to choose from, A, G, C, or U, and you only get to pick 2. And then you can stitch those 2 together to make a repeating sequence, say up to 12 repeats. And then the object of the game is to find the combination that will give you either the most compact fold, so you get a point for that, or produce the most distinctive or unusual structure, you get a point for that, or produce the greatest number of different structures. And so everyone can choose. There's only 16 possibilities here. We can have AA, AG, AC, or AU, or GG, GA, GCG, etc. And then as I go through the talk, you can see if your combination you think is still a good choice, or maybe you want to change. Maybe I can change your mind. I don't know. I just made up this game yesterday. So I think the Build-a-Cell theme kind of got me thinking. I'm not a synthetic biologist. I'm an RNA biochemist, but I love to think of things like the origin of life. And I know Kate thinks about this kind of stuff a lot. And what I think, and I think most scientists believe now, what must have happened around 3.5 billion years ago is that a membrane-bound compartment containing self-replicating genetic material must have formed. And so it probably began with very simple chemistry and gained complexity over time with evolution. And if we think about this genetic material, that would need to assemble and fold up so that it would have some activity and be able to replicate itself. And it also have to probably unfold to serve as a template to make more of itself. And there are various number of groups now that are trying to, reverse engineer such a scenario. I just found a recent paper on this. It's pretty cool. It uses an RNA that stitches together trinucleotides using freezing and thawing to make copies of RNA. So it seems like a plausible thing that may have happened at one time. But if we go, ahead in time to modern day, we see that about 3% of the human genome is simple sequence repeats. That's more than the number or more than the percentage of the genome that codes for proteins. So there's a lot of repetitive elements. The most common simple sequence repeat in human DNA is GT and AC repeats. So just GT, 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 and AC, 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 where N is the number of repeats. And these are equal to one another in DNA, of course, because we have complementary strands. And we know that the quantity of G equals C and A equals T, et cetera. So those are always going to be equal in DNA. But in RNA, that's not true. So the GT repeats tend to be downstream of promoters, and the AC repeats tend to be upstream of promoters. So by far, the most common simple sequence repeat in human RNA is GU or UG. And so I've written 2 sequences here. a G repeating sequence and a UG repeating sequence. And what you can see here is they're really the same sequence, and one is just missing a 5' nucleotide. And so collectively, what my lab calls these RNAs are PUGs, or poly-UG. But PUGs can start with a G or a U, and they're exactly the same thing. It just depends on the length. And humans have many of these sequences. And where I got interested in this was in 2019, a colleague of mine, Marv Wickens at UW-Madison, discovered a poly-UG polymerase from C. elegans. This enzyme's called RDE3, and it synthesizes PUG tails on RNA 3' ends. The way that he found this was through a high throughput screen, and he basically hypothesized that there must be some ribonucleotidyltransferase enzymes we don't know about. The most common one that we think about in terms of these types of enzymes is poly-A polymerase that adds poly-A tails to RNA 3' ends. But this is a super family of enzymes, and there are many genes that are homologous to poly-A polymerase, especially in other organisms that look like poly-A polymerase but had not been characterized. And so he did a high throughput screen, and he found that C. elegans has a poly-UG polymerase. Moreover, that polymerase had been studied for a while, and nobody knew it was a poly-A polymerase. So Marv's lab proved this by tethering the enzyme to an RNA and watching it make these poly-UG tails, which could be either poly-UG or poly-GU, meaning the enzyme initiates equally well from a U or a G, and the tails could be 100 nucleotides long or more. This was interesting because this enzyme, RDE3, was previously shown to be required for RNA interference, which was discovered in C. elegans. RNA interference is a type of innate immunity, and humans do RNA interference too. They have slightly modified pathway that goes through microRNAs that then get processed into siRNAs. C. elegans has a lot of the same machinery and has evolved some different activities. One of them is this PUG polymerase. So interestingly, if you look at these PUG sequences, we see that humans have many thousands of PUG repeats encoded in their RNA transcriptomes for the reason that I told you before. The human genome is full of simple sequence repeats. GT is the most common one. It's just equal to AC, but in RNA, GU repeats are prevalent. There's thousands of them. That's probably because DNA polymerase slips. And so once you get a few GTs, they have a tendency to expand and get inherited. But honestly, we really don't know what these simple sequence repeats are doing. But if we compare that to C. elegans only has a few poly-UG or PUG repeats in its genome. And that's because they're poisonous. And I'll tell you why now in the next few slides. These PUG tails induce gene silencing and transgenerational epigenetic inheritance. This was discovered in 2020 by Scott Kennedy's lab at Harvard. And what Scott did was, he's a C. elegans biologist, fantastic scientist who studies RNAi, and they can inject live C. elegans. And you can genetically manipulate these animals, in this case, to express GFP in the germline. And each one of these green circles that you're looking at is a developing embryo. So the worms are hermaphrodites. They self-fertilize their own eggs. These embryos start developing, and it has a germline expression of GFP. And these worms are a mutation in a pathway of RNAi, which goes through double-stranded RNA processing through an enzyme called dicer. So this was a control experiment to show that this activity is something different from the canonical RNAi pathway. They inject in double-stranded GFP RNA, and there's no effect really. You can still see the GFP expression. Again, these are live animals. But if you take a fragment of GFP and you add a pugtail to the end of it, the gene silencing is rapid and almost complete. And this is a sense fragment of GFP, around 200 nucleotides, with a pugtail, in this case, 18 repeats of UG. It doesn't work with any other repeat. An interesting thing about this is that this gene silencing that happens with the pugtail RNA is inherited. So just a single injection of the adult, the GFP expression turns off. The progeny of that adult, or the F1 generation, if you will, also has most of the GFP turned off. It takes 7 generations for the GFP to fully come back. So that's called transgenerational epigenetic inheritance. And what it means is that the adult animal was somehow able to package these pugtails into the embryo, and then they somehow amplified that and kept creating these pugtails in the next generation. And over time, that effect is lost. But I thought that was like a really fascinating thing. No one had ever observed this until 2020. And so now you can see why the C. elegans genome is almost devoid of pugs, because they turn off gene expression for generations. So what C. elegans uses these pugtails for is as a form of innate immunity to combat viral infections and also harmful genetic elements like retrotransposons. Or in this case, GFP. So it's a portable type of system that lasts for generations. So here's the model that I've sort of recartooned from Scott Kennedy's paper in 2020 for how this might work. So I told you there's an enzyme that adds poly-UG tails. You might be thinking, how does an enzyme know how to add poly-UG tails? It's easy to think about, adding poly-A tails, because that enzyme can only take ATP and start adding that to the end of an RNA. But how does it know how to add alternating nucleotides, always repeating either UG or GU? We don't know. We don't have a structure for this enzyme, but there's precedence for this. The tRNAs have enzymes that add CCA to the end. That enzyme uses a single active site. And as the RNA chain grows, the size of the active site grows and it switches from C to A. And I imagine it's a similar mechanism here, but we don't yet know. Regardless, that RNA adds pugtails. And I just showed you an experiment where they attach a pugtail to a GFP. That pugtail then recruits, it's a signal for recruiting a very specific enzyme in C. elegans, which is an RNA-dependent RNA polymerase. It lands on this pugtail and starts making siRNAs. It's basically marking, the pugtails are marking the RNAs as templates for siRNA synthesis. Those siRNAs now are complementary to whatever RNA this is, a viral RNA or GFP, for example. They can go and cut other RNAs. Those other RNAs can get repugylated. So all of these enzymes talk to one another and are probably in the same complex. They form in a foci. There's been some work done on that. We don't know what the complex looks like, but Scott calls it a pugosome. And what happens is that the RNA gets cut, it gets repegylated. That means you can then make more, and this is sort of an amplification cycle. So if you inherit a pug RNA, the progeny can do this, and then they can repackage. And over time, because this involves cutting, the RNA gets shorter and shorter and shorter, and after about 7 generations, it can no longer act as a template. So that's the model. And so this is where I got interested in this. So we've been working on this now for almost 5 years. The first question we asked was a very simple one. We asked, does the pugtail fold into any kind of structure that might be required for, say, recognition by this enzyme, this RNA-dependent RNA polymerase? And so to do that, we just made a bunch of pug RNAs. This one has 10 repeats. And this is a circular dichroism experiment. It looks at RNA folding. It looks at RNA secondary structure content. And this is almost a flat line. So this is really sort of unstructured. If we go to 11 repeats, we start to see some features, but it's not stable at room temperature. There's still not very much structure. But if we go to 11.5 repeats, now we see these huge signals here. This is a folded RNA. And moreover, it's a really unusually folded RNA. So like this peak at 260 is characteristic of right-handed A-form helices. But this peak over here, this is really strange. This peak at, this negative peak at 304 nanometers. That's been seen only once, as far as I can tell, in any nucleic acid. And what that is indicative of is a left-handed helix. It's called a Z-form helix. Okay, so we see no structure at 11 and structure at 11.5 repeats. That means 12 guanosines, 11 uridines. If we then make it longer, structure is still there. If we flip it from GU12 to UG12, the structure is still there. And that's what we'd expect. The only difference between UG12 and GU at 11.5 is that this one has an extra 5' U. Okay. If we put it in the middle of an RNA, we still see the structure. So it doesn't have to be at the 3' end to form a structure. And if we put it in lithium, the structure goes away. And that's characteristic of a quadruplex. So all of these are folded in potassium. So when I saw this, I emailed Scott Kennedy at Harvard, and I said, hey, Scott, we see something really cool here. And I predict that, like, if you had a pugtail of, say, 11 repeats, that would not be able to silence a gene. But if you have 11.5 repeats, it will, because we see this really dramatic change from a single strand to a highly structured RNA right at that threshold. So, and I sent this data to this guy, said, okay, we'll do that experiment. And so here's the results. This is a multi-generational assay where these worms have a lethal gene that they, temperature-sensitive lethal gene that they pass along to the progeny. The progeny will be dead unless that gene is silenced. And if they saw that at 10.5 repeats, all the progeny are dead. And at 11.5 repeats, which is right where we see this RNA starting to fold, now they start surviving. And even better at 12.5, 13.5. So when we saw this, we immediately thought we were onto something, because it's really rare that biochemistry in a cuvette would agree so well with a live animal assay. So we thought, okay, maybe this RNA is really unusually structured. And maybe that's the signal that recruits this RNA-dependent RNA polymerase to make siRNAs. And so we did a lot of work on this. I'm just going to show you one example of how we proved that this RNA was folding in a live animal. And that is, we knew it was a quadruplex. I showed you it's unfolded by lithium. Also, the CD was characteristic of quadruplex. And so we could make, or basically just buy, atomically modified RNAs, 7-deoxyguanosine, which no longer have this N7 nitrogen. They have a carbon. And that knocks out 4 hydrogen bonds in a G quartet. And we can show that when we run an RNA on a native gel, it folds into a pug fold. If we look at a single-stranded control like AC12, it runs a single-stranded RNA. And if we do the diaza, it also runs a single-stranded RNA. If we inject, now we can ligate this on to GFP, which we did, and we sent that to Scott's lab, and Scott injected that GFP, well, first a control GFP, where we just ligated normal RNA, normal pug onto there, and that's silenced. And then the experiment where we ligated on the 7-deaza, which knocks out this quadruplex, and that does not silence the GFP. So directly demonstrating in a live animal that you need a quadruplex formation in order to do gene silencing. And I think this is an important thing because it's really hard to prove a structure in vivo. And I think this is one of the first times that quadruplex has been really shown to be functionally important in a live animal model. So here's our refined model now, is that you add a pugtail to targets. Again, we don't know exactly what's targeting this system. There's a lot of biology here that's not understood. But these pugtailed targets are viral RNAs and retrotransposons, as I mentioned. They have to have at least, they always have to have 12 Gs in order to be active. And they fold into a quadruplex structure that is recognized by this RNA-dependent RNA polymerase. And we've done a lot of things like pull downs to show that's the case. Again, with the N7-deaza, doesn't pull down the enzyme, et cetera. So this is our model for how this type of gene silencing is working. We wanted to know what the structure was. We crystallized GU12 and GU11.5. Both of them active for silencing. Both gave the same crystals. They diffracted to 1.97 angstroms. The crystals are red. They're really cool looking. That's because we had to add a ligand, which is a porphyrin. This is N-methylmesoporphyrin. And that just got the RNAs ordered, and it acts like a cushion in between the quadruplexes to help them stack. And this is what the crystal structure looks like. It's indeed a quadruplex. There are 4 layers. Here's the porphyrin up on top here. I'll show you the different layers of the structure separated out here. And so here you see the porphyrin ring stacked on top of a G quartet here. 2 potassiums in the middle and also on the edges. And then there's another G quartet and a G quartet and a U quartet. And there's some bulged out U's. I'll show you a little bit better picture of what this looks like. We needed to be sure that somehow the porphyrin wasn't influencing the structure or making it look different. So we also did NMR. Most people would not want to do NMR of dinucleotide repeat that's 24 nucleotides long, because you would think that all the signals would pile up on top of each other because there's no diversity to this sequence. But in fact, all the Gs are separate, which was a surprise. So we can resolve them all. That's because they're all in different conformations. Each layer of the structure is in a different conformation. So, and we can even walk through and map the hydrogen bonds. I'm not going to go through these experiments, but it's a really cool thing we can do by NMR is that you can directly detect the hydrogen bonds. So here's the structure. The short story is that the NMR structure is exactly the same as the crystal structure. And so here's the sequence here, 24 nucleotides of GU. If you have, so that's called GU12. If you have UG12, what you do is you end up putting an extra U on the 5' end and then going through the same structure here. And so the structure starts here. Here's a 5' end, and that has a quartet here. We'll call the G1 quartet. This quartet is in a conformation called syn. That's a glycosidic angle, and 2' endo. That's a sugar pucker, as you know. And then we have a bulged U here. That's this U2. And then we have an inversion of the backbone. This G is upside down relative to this G, and it's in a different conformation. It's syn C3' endo inverted. Hence why these have very different chemical shifts in NMR. Even though they're both G quartets, they're G quartets in totally different conformations. And then what we have is a big stretch here from G3 down to U4, which is at the edge of the structure. And that's here. And this is a U quartet. This U quartet is anti-C3' endo inverted. And then we have the backbone curls, and then the G5 quartet is interleaved between 3 and 4. So the stacking is not sequential. The stacking order is 1, 3, 5, 4. And this G5 quartet is anti-C2' endo. Again, totally different conformation. So in fact, if you wanted to mix and match glycosidic torsion angles and sugar puckers, we have every single possibility you can think of here. So it's a simple structure with a fairly complex fold. And this was the first time I've seen this, that you can actually have so much structural diversity in such a simple sequence. And moreover, this is the most compact fold you could imagine. It folds around a core of metal ions, potassium ions, and these give it a lot of stability. Its melting temperature is around 53 degrees Celsius. Not hugely stable, but certainly stable enough to form in vivo. And you really couldn't imagine squeezing RNA into a more compact volume than this. So we have structural complexity encoded by a simple dinucleotide repeat. So that was a surprise. In fact, I've just walked you through the structure, but even if you were to know what the structure looks like, and I said, find the 4 uridines that make the U quartet, you probably have a hard time looking at the sequence. In fact, these are the 4 boxed here. Only these 4 make the U quartet. All the others are bulged out of the structure, either as a bulge or as what we call a propeller loop. Single nucleotide spanning 4 quartets here, because of the backbone inversion allows that. So how does an RNA know to form the specific structure? The answer is we don't know. We are still not smart enough to be able to predict RNA structure, even though AlphaFold 3 has this access to this structure. It cannot predict the structure, meaning AlphaFold 3 came out last year. This structure I'm showing you came out in 2022. It was in the database, sub 2 angstrom structure. Apparently, it didn't make the training set, because if you give this sequence to AlphaFold, it cannot predict this structure. So we need better tools for understanding RNA structure. This is what we call a Z-form quadruplex. What do I mean by Z-form? Z-form is left-handed, and this Z-form structure here on the right is CG repeats. And it forms a duplex because CGs pair in a stable Watson-Crick fashion. I'm just showing you one strand. But this duplex is going the opposite direction of a normal right-handed duplex. You know how to do this with your right hand. And the A-form direction is that all the sugars are pointed the same direction, 5' to 3', and all the stacking is right-handed. Whereas in the Z form, what happens is that there's syn-anti inversions where the sugars are pointing towards each other, away from each other, towards each other, which is what I'm showing with these arrows. So these two sugars are pointing towards each other, and these two in the middle are pointing away from each other. It's alternating syn-anti, syn-anti, and that's exactly what this PUG fold is doing. It's repurposing the Z form backbone pattern into a quadruplex. And so I think simultaneously what we've been able to learn here, first of all, is that quadruplexes don't need to have any repeating Gs. This has no sequential Gs. It's GU, GU, and it used to be thought that quadruplexes only happened when you had like GGG, then some couple nucleotides, and then GGG. You don't need that kind of repeating sequence. Second of all, we now know that Z-form backbones can fold in vivo, which wasn't known before. At least hasn't been proven directly yet. But now we know that we can form these kinds of structures in vivo. The Z-form structure, the duplex of CG repeats, has been somewhat controversial because that only forms a crystal in 1 molar salt. That's because the backbone is only about 8 angstroms apart. The PUG quadruplex fixes that by taking ions into the center and moving everything apart and forming quartets. So it solves the electrostatic problem, and it can form in very low potassium, like 5 millimolar still. I told you that humans have a lot of these sequences embedded in our own transcripts. These PUG sequences here, 12 or more repeats. How many of those are in the human genome? If you just map it to the human genome, you find 40,000 examples, about 20,000 in genes. Sorry, you find 40,000 outside of genes and 20,000 inside of genes, so 60,000 total. That's a huge number. That's way more than you would predict if sequences were random. They're not random. And in fact, these GU repeats are not even randomly distributed. They happen to pile up near splice sites, either 5' or 3' splice sites. So this is just mapping where these are in the transcriptome on a generic sort of intron-exon boundary. And in fact, if you look for other repeats, I told you that AC repeats are equal to GT in DNA, but not RNA. So there's fewer ACs in RNA compared to the blue. These are the PUG repeats. And these are randomly distributed, whereas the PUGs are not. AUs, there's even less. So these would be 12 AU repeats compared to 12 GU repeats. Again, they're random. If you go through, every other dinucleotide repeat is randomly distributed, except for the PUGs. So that's a little thought-provoking. Why is that? We honestly don't know, but these PUGs have been linked to a lot of diseases. They tend to be polymorphic, meaning that some people will have a certain number of GU repeats encoded within their genome, and other people will have either longer stretches or shorter stretches. These GT polymorphisms have been known for a long time, and through GWAS studies, just genome-wide association studies, they've been linked to diseases like here's colorectal cancer. These are just called GT polymorphisms because it's just looking at genomic DNA. Nobody really had ever proposed that they could actually be working at the RNA level, except for in one case, if you go all the way down to the list, there's a cystic fibrosis case here. This has been clearly mapped to an intronic sequence that causes exonic skipping in the cystic fibrosis transmembrane conductance regulator gene. This is a rare hereditary version of cystic fibrosis. Normal individuals have 11 UG repeats next to an exon. and people with 12 repeats get exon skipping. And that's exactly where we see the boundary for the pug fold. Is it coincidence? I don't think so. But we haven't gone back to map. We're working on mapping pug folds in human cells now. So it could be that these RNAs fold up in some cases in a way that's unproductive in human cells. Tom Check's lab, right after a year after we reported this structure, found an enzyme that binds to it. This is DNMT1, DNA methyltransferase 1. This enzyme silences DNA. What is it doing binding to a pug-fold quadruplex? Again, we don't know. There's a lot of biology here that has to be figured out. Humans have a lot of pugs, and they have proteins that bind them. What happens when they're long? Humans have about 2,000 pugs that are more than 24 repeats long, so really long pugs. A famous one is in the long non-coding RNA MEAT1, which is a cancer-associated transcript, has huge number of pug repeats. This causes multiple pug folds to fold like beads on a string. We can do that. We can measure this by CD. We can get fraction folded, basically, of the RNA by comparing different lengths. And as the length gets longer, you get more single-stranded, and the secondary structure content goes down until you get above the potential to get 2 repeats. It starts to go back up and becomes more folded. Fits perfectly on a line. That's 2 pug folds. You can also do this on a gel to compare lithium versus potassium for different lengths, and you can plot them. And once you get to 24 repeats or more, you start to see 2 bands. And that's because these RNAs are folding randomly, either 1 pug fold in the middle or 2 pug folds on the end. You can nuclease map them, and you see the 2 pug folds in the middle. If they're separated at all, then this is RNase T1. It will cleave them, and you end up with just a single pug fold. So these can form like beads on a string. We connect our crystal structure. We see, we get something like this, but if we run it through MD simulation, they stack on each other and remain stable. So, and now you have room actually for a third pug fold to form. Okay, what about short pugs? We started wondering just the general behavior of pugs, and we saw something. We did a control where we took some that were too short to fold up into what we call the pug fold. This quadruplex needs 12 Gs, as I already showed you. So in potassium, what that happens to do is it forms these condensates. These are micron-sized condensates. We put in lithium, they dissolve. Because these quadruplexes are all stacking on top of each other and forming a very condensed phase. But we saw this happen even with short pugs. So something's weird here. Like, how could these short pugs be doing the same thing? The structure has 3 G quartets. You need 12 Gs to do that. We found so even really short RNAs like GU3 make these condensed structures. And other G-rich RNAs did not. So like here's GGU. That's a 6-mer. It doesn't form a condensate. This is GU. This is a 6-mer. It does form condensate. This one has more Gs than this one. So it's not just a property of Gs. There's something special about pugs in general. And in fact, you can see that these condensates are gels. You can frap them, hit it with a laser, and they basically don't recover. That means they're not very liquid-like. So there's GU12. You can do the same thing for GU18. Hit it with a laser. It doesn't recover. And these are fluorescein labeled. But if we have the unlabeled RNA, we can still form the condensate. Now we can add N-methylmesoporphyrin to it. And we can frap that because that's a fluorescent ligand, and that comes back right away. So what that means is a small molecule can diffuse on and off to these condensates, but the RNA inside is not really moving. So what does this look like? Maybe something like this. If you zoom in from the micron scale down to the angstrom scale, we have packing of quadruplexes. The crystallite, so we did some CD on these short quadruplexes. I'm running out of time. I'm just going to tell you the punchline here. So if you have a really short quadruplex here, GU4, compare that to GU12. These two CD spectra look like inversions of each other. They're almost mirror images. So you see a positive peak here, negative peak here, negative peaks here, positive here. And so what this is a right-handed quadruplex, and this is, as I've already showed you, is Z-form left-handed quadruplex. These are intermolecular, these short GU quadruplexes, these right-handed quadruplexes. They have KDs around hundreds of micromolar they need to form. That's because they're highly concentration dependent because you need 4 strands to come together and they're short. So this is what the crystal structure of GU3 looks like. This is a 1 angstrom structure. There are 8 strands here. This is 2 intermolecular quadruplexes stacked on top of each other. So if you take a slice, what you can see is 2 strands of GU stacked on top of each other. And these two here's a monomer down here. So these two quadruplexes are not the same, actually. They're structurally different. For example, one has a water coordinated here, and this one does not have a water coordinated up here. So that's a little bit surprising. So we have an empty quartet on one side of the structure and a water-mediated quartet on the other side. Symmetry related. But 1 angstrom, it's really unusual to see structural heterogeneity because usually that causes crystals to become less ordered and lose resolution. But in fact, in this crystal, what we can see is actually there's multiple conformations for each strand of the RNA. Frozen snapshots of dynamics, if you will. So for example, at this GU step, we see 2 phosphates equally populated. What that means is that the crystal has 2 conformers within it that are roughly equally stable, and they froze like that in the crystal. So we have, again, this is a right-handed quartet now. It's sort of the opposite-handedness of what I showed you before. And again, each layer is different. So we have different types of quartets, U quartets, G quartet, U quartet. And even the U quartets here are different kinds of U quartets. We have an anti-C3' endo and an anti-C3'. And so comparing these two, these have exactly the same number of nucleotides now, and they're also exactly the same type of repeating sequence, UG. The only difference is that this one is 4 strands of GU3, and this one is GU12. This one is intramolecular, left-handed, very compact. This one is intermolecular and right-handed. So you can see how the backbone curves to the right here, and this backbone's going to the left over here. And if you look down from the top, you can see this one's going counterclockwise, that's the left-handed, and this one is going clockwise, that's a right-handed, like most helices. So that's the end. Conclusions from this is that quadruplexes do not require consecutive Gs. We learned that. We also learned that they can incorporate Z-form backbone conformations. We learned that the pug fold, this unusual quadruplex, directs RNA amplification in C. elegans. Functions in live animals. Humans have many thousands of potential pug folds, around 20,000 sequences. that are capable of forming a pug fold. In some cases, they've been linked to disease. These even short pugs can self-assemble into condensates when crowded. The short pugs form right-handed poly-UGs. What I didn't have time to tell you is that this fold does not have to be poly-UG. So we started asking, what are the rules for these pug folds? It turns out you can change the U's. Most of the U's are not involved in the structure. Those can be just about any nucleotide. So there, in fact, are way more than 20,000 potential pug folds in the human transcriptome. These are the people in my lab who did the work. Scott Kennedy, our collaborator at Harvard, Marv Wickens, my colleague who discovered the pug polymerase. And I thank you very much for your attention. Happy to take questions.
Thank you so much. There is several questions in chat. Can you see chat?
Let's see. I'm gonna, oh yeah, I
can read the questions to you too if it's hard to see them.
Okay, yeah, I lost my my toolbox somewhere.
All right, I'll start from the top. Okay. Daniel's asking, how is the PUG polymerase dealing with possible quadruplex formation creating stable folds? I thought it's very difficult to read through sequences like that.
Great question. Yeah, so it doesn't have to read through the sequence, but what it has to do is create the sequence. But you might, like, I think as you're alluding to, it's a great question because as the RNA folds, that might interfere with the polymerase. And we know of examples like in bacteria where RNA hairpins pull polymerases off, et cetera, and act as terminators, right? And maybe a quadruplex could do the same thing. That's a cool question. And one answer I can give you is that we have a paper that's in press right now at Nucleic Acids Research, where we mapped how long it takes for a pug fold to form. It's a Z form and it has a lot of backbone inversions and syn-anti. It takes a long time for the RNA to fold, actually. It takes about 15 minutes for the RNA to fold. So like a hairpin, that takes like a microsecond to fold. Hairpins can snap together really quickly. Quadruplexes are slow. So, but then how fast is the polymerase? Hasn't been reported yet. I know of some unpublished data that says that the polymerase is also slow. So that could be because we're missing a plasticity factor or something. But the RNA is really slow to fold. So in C. elegans, maybe the polymerase makes it and then it takes minutes for the RNA to fold. It doesn't really matter because it takes, hours to days for the embryo to develop, so it can still be inherited.
Thank you. Okay, next question from Javier. Did you ever look at PUG folding and quadruplex folding in general in cell-free systems? Would the silencing effect persist in transcription in vitro?
Yeah, no, we haven't done that. That would be amazing, actually. I don't, I mean, maybe you know more than I do, because I've just gotten into into reading about RNAi and stuff, but I don't think that we have an in vitro system even for simple gene silencing. Like let's say, maybe Kate, maybe you know this, but let's say we have in vitro translation systems. Has anybody taken, say, like the machinery needed to synthesize a microRNA, process a microRNA, load it into an Argonaut and silence that in vitro? I don't think anybody's, fairly complex
No,
pathway. Yeah.
as far as I know, it hasn't been published. I don't know if anyone tried and failed. I've never seen any reports on that.
Yeah, I think the easiest way would be like if you just made a pre-processed siRNA and loaded that into the Argonaut and then gave that complex into an in vitro translation system, that might work. I don't know if anyone has done that. I think we're far from being able to assemble this in vitro, but that's obvious. That would be an obvious goal to really understand and dissect the system. I think that would be fantastic. We're just
Yeah.
sort of just baby steps right now and trying to understand how this works.
Yeah, I think the next question kind of touches on that too. Next question is from Angela. Interesting talk. This could be a valuable gene regulation mechanism in simple synthetic cells. Do you know if anyone ever tried to use PUG or other G-rich sequences to intentionally regulate gene expression, like engineer it to have specific expression rates different than untagged sequences?
Interesting. I do know of one study where so people have looked at the ability of quadruplexes to regulate ribosome loading or scanning onto messenger RNAs or 5' UTRs. And I'm going to not remember, this is a very recently published paper where quadruplexes can slow down that process or inhibit that process. And they looked at pugs in that particular case, and they didn't see that it slowed down, I think, the translation. But then they added a ligand that bound to a quadruplex binding ligand, and then they saw it did. So the idea was that it could be a programmable system, but you need to stabilize it with a ligand. Like I showed you one ligand, like N-methylmesoporphyrin that sits on top. The pug fold is not as stable as some quadruplexes have TMs that are like close to the boiling point. And so they're really stable. And these canonical sort of quadruplexes have like GGG, NNN, GGG, NNN, GGG, et cetera. And those are stable, and those seem to be able to regulate the loading of ribosomes. The pugs, not so much, maybe unless if you give them a stabilizing ligand.
Mm-hmm. I think I remember one of the papers you mentioned. There was a paper, I think 3 years ago, from the Balasubramanian group where they did the quadruplex ribosome speed of translation over some kind of a weird motifs.
Uh-huh.
That might be that.
Yeah. Yeah,
Okay.
my memory for citing literature is I can't do it without PubMed or Google search anymore.
Yeah, me too. We lost the functional memory. Okay, next question is from Rosalinda. Are pugs eukaryotic only, or do we find them in prokaryotes too?
That's a great question. I'm sure they are in prokaryotes. I don't think they're as abundant in prokaryotes. Haven't done a genomic analysis of them in prokaryotes. That's probably something that we should do. That's a great question. In humans, there's a couple of things that are loading up the human genome with pugs that bacteria don't have as much. One is something called CPG suppression, which is cytosines get methylated. The methylation of cytosines causes a spontaneous deamination, and then that C can be misread as a T. So the cytosines that are methylated are in CpG repeats. And so these can lead to GU repeats over time. And I don't think bacteria have that same way of acquiring GU repeats that eukaryotes have. But certainly they have polymerases that can slip and they have repeating sequences. I just don't know the frequency.
Mm-hmm. And question from Tom. Are POG fold obeying all the same folding rules as regular quadruplexes? For example, are they lithium dependent?
Yes, they're unfolded in lithium, but they're different from other quadruplexes. One thing we know is that they're highly specific for potassium. They don't fold in sodium. So some of the other canonical like DNA quadruplexes that have been studied will fold in potassium or sodium. The pug fold is really selective for potassium. It doesn't fold in sodium at all. But we also know other quadruplexes will form if they're DNA or RNA sequences. So like the canonical right-handed GGG quadruplexes can be DNA or RNA. The pug fold can only be RNA. It doesn't fold in if the DNA form, the GT repeats don't form this quadruplex, and we don't know exactly why yet. So there are some peculiarities about the pug fold, and that it really needs potassium and it needs to be RNA.
Yeah. Thank you. I think that's all the questions. I want to say I was actually thinking myself about the Argonauts a while ago. Could we do that in vitro? It seems I don't see like an obvious reason why we shouldn't be able to do it, but I don't think anyone ever tried.
Yeah.
It would be
Yeah,
cool.
I don't know. Yeah. But thank you very much for this opportunity. Yeah,
Thank you.
yeah, and yeah, if we want to chat more, let's stay in touch. Maybe we can think of some cool experiments together.
Absolutely. I'll send you a follow-up email. I usually do.
All right.
All right. Thank you so much and have a great rest of your afternoon.
You too. Thank you, everyone. Bye.
Bye.